Hello, I'm Ariadna Bartolo and I'm going to talk about immunogenicity induced by our academic CAR-19 in patients with relapsed refractory B-cell malignancies. CAR-19 cells, also called ARI-001, is a cell therapy approved by the Spanish Agency of Medicine in 2021 for the treatment of relapsed refractory acute lymphoblastic leukemia. This academic CAR-19 consists of the anti-CD19 single-chain variable fragment originated from a mouse monoclonal antibody A3B1 and conjugated with the co-stimulatory domains for 1BB and CD3, which are fully human. Its safety and efficacy have been demonstrated, but no studies have been conducted about the immunogenicity. So, our main objective is to assess the immunogenicity of this academic CAR-19. First of all, we assess the presence of human anti-murine antibodies called AMA in patient sera by a cell fluorescence assay. This assessment was carried out at different time points. Then we perform cytotoxicity assay for AMA-positive patients to appraise the effect of these antibodies. And regarding to the cellular immunogenicity, patients included in the clinical trial were HLA typed, and then we predicted in silico the binding of the CAR-19 peptides into these HLA molecules. A total of 47 patients were treated with this academic CAR-19, and approximately 25% of these patients tested positive for the presence of anti-CAR antibodies after 3 months of the first or second infusion of ARI cells. Even though 25% had anti-CAR antibodies, only some of them were diminishing the cytotoxic effect of CAR-19. In relation to the prediction in silico, a set of 94 priorized immunogenic peptides of CAR-19 were selected for assessing the T-cell response. In this case, 62 for HLA class 1 and 32 for HLA class 2. Finally, our academic CAR-19 induces humoral immunogenicity in 25% of the patients. AMA diminishes the efficacy of CAR-19 in vitro, and this data also suggests that the assessment of humoral immunogenicity could be important in patients who need a second infusion. Finally, the most immunogenic peptides are in the single-chain variable region. Thank you.